Okay, so this is going to be the recording for the part two, the part B of the vision chapter, the special sense chapter, where specifically we're just covering the vision aspect of it. But part two is the one where I wanted to make the video because this is more physiological, less microscopic anatomy. So this is really just going to be the photo transduction, and then also like the visual pathways, things like that. So this is not exactly rehearsed. I skimmed through the PowerPoint, but let's hope I do okay for the first run through. Alright, so let's just start this here. Alright, so for the first thing is photo transduction, the process by which light is converted into electrical signals. These will be in graded potentials via the photoreceptors and the ganglia in the retina. So if you recall, remember ganglia is just a cluster or a large group of cell bodies within the peripheral nervous system. So for example, when you look at a portion of the retina, which maybe this will be fine right here, but the retina is located all throughout here, and when you basically zoom in, you see many cell bodies, right? So basically the ganglia itself is just a cluster of cell bodies within the peripheral nervous system, so you're going to have the photoreceptors, also many cell bodies within this area. Alright, so back to here. And keep in mind the photoreceptors themselves and also the bipolar cells only do graded potentials. The ganglion cells are the only ones that do the action potentials, okay? When looking at the photoreceptors themselves, right, the portion of photoreceptors are embedded into the pigmented layer. So if we look right here, this qualifies as a photoreceptor, and also this one right here. These are just the rods and the cones. And they're shaped because where they are embedded into this pigmented layer, right, this is one pigment cell, basically the shape of their outer segment is basically distinctive towards their name. So this is rod shaped for the rods in the outer segment, and then the outer segment of the cones is cone shaped. Okay, but basically only the outer segment is embedded into the membrane of the pigment cells or the pigmented layer. When you see the inner segment, it's not embedded into that membrane. So the outer segments are the receptive structures. The outer segment joined to the inner segment, which is farther from the choroid layer, which joins the cell body. So let's look over here. This is the pigmented pigment cell right here, and then the choroid layer is actually beneath here. So as you go towards the inner segment, you're always going to be obviously going away from the choroid itself. And as you go away from the inner segment further, the cell body is even further from the choroid. Okay. And opposite to the outer and inner segments are the synaptic terminals. So you can see opposite from the outer and the inner segment, and from the cell body, you finally see over here, which are the synaptic terminals. And these are just going to synapse with the bipolar cells right after. Okay. I guess that really covers this entire slide here. We'll see a little bit about this part, and I'm not sure exactly which slide, but this I'll explain once we get there. Alright, so presence of visual pigments in photoreceptors. So this says we have 3 visual pigments that exist in humans. Really we have 4. Okay, I think what they're saying here is the visual pigments for the higher wavelengths of light for that the cones are responsible for. Okay, and the cones are they basically have these 3 visual pigments themselves. Okay, they have a visual pigment for red, for blue, and for green. When we look at the rods, they have another visual pigment that basically absorbs at much lower wavelengths, but there really is 4 distinct different types. 3 for the 3 visual pigments for the cones, and then 1 for the rods. Okay. Now, rods and cones have visual pigments in their outer segments. Okay, rods can have longer outer segments, and then the cones have the shorter outer segments. So we can see over here, obviously the outer segment for the rod, it's much longer, and then the outer segment for the cone is much shorter. But when we look at the outer segment, specifically here, is where we see these things. They're called discs, okay? And these discs actually contain the visual pigments. And just quickly, I want to see if they have this structure here, which are the discs for the rods. They're just simply taking this structure here from anywhere along this outer segment. So we'll say we'll take away at least 3 discs here, and then we zoom in. Here is where you can see that it is a membrane, just like the membrane on any cell, the phospholipid bilayer. But embedded into this membrane is where you see these visual pigments. Okay, so for example, this visual pigment here is saying it contains these 2 components, and this is actually the visual pigment for the rods that I was speaking of. But once we get there, we'll explain that a little bit more. Okay, regeneration of the outer segment. So these discs that we're talking about are replenished almost daily, almost every 24 hours, because although they are the retina is made to basically convert the what's called the light energy into the electrical signals. They're very sensitive to this light in the first place, and they can get damaged relatively easily. So what happens is this outer segment here, the discs that are located in the outer segment, what happens is they are replenished about every 24 hours. And what actually happens is, this is not really much for memorization, but because they are saying that there is regeneration of the outer segment and these discs are replenished almost daily, it really just has to do with these discs being sent to the tip here, and then basically fragmenting off, and then basically they get phagocytized by these pigmented cells, and then new discs are basically regenerated at the cell body, and they just send it down. But basically, because of this regeneration, yes, there is very easily damage done to the retina almost on a daily basis from, like, let's say, high intensity light. But because of this system we have, we're able to regenerate the most important portion, which is the outer segment, which contains the actual visual pigments. Okay. We just looked at this picture right here. So this is one example of a visual pigment. So the visual pigments are composed of retinal and opsin. So this is one visual pigment. This portion, the little beaded portion here, is a representation of the retinal, and the retinal is actually the light absorbing molecule. Okay, while the opsin, which is this highly folded protein portion that you see here, is the just the protein portion. Okay, it does not do any of the absorbing of the light. That's specifically done by the retinal. Okay, now the retinal is derived from vitamin A. Okay, and then several isomers exist. So for example, if we're looking here, for example, so how do we get, don't worry about this right now, this is just one of the isomers we're going to talk about. This is what we're talking about right now, the retinal. Okay, so it's derived from vitamin A, right? So basically we get the vitamin A from the blood, and what happens is there's a process called oxidation, and then we're able to convert the vitamin A into our retinal. Okay, we're going to go more into detail in a little bit about this, but basically just important to know that the retinal is derived from vitamin A. Okay, so vitamin A is incredibly important for eye health. Okay, continuing on, several isomers exist. For us, we're just going to be considering just 2 of the isomers. Okay, what we're really looking at in one example of what happens in rhodopsin, really here is we're going to look at the 2 isomers, which is in the dark. We're going to have something called 11-cis retinal. So that is one of the isomers. And then basically once photons are absorbed, once light energy is absorbed, there's this basically series of steps that occurs in which the retinal will detach from the opsin, but then we get the other isomer here, which is called all-trans retinol. So the two isomers you guys are responsible for is the 11-cis retinol, which is while in the dark, as you can see here, and then the all-trans retinol, which occurs when in the light, when light energy is absorbed. Okay, so the opsins, you have different forms of protein associated with the retinol. So you don't just have one type of opsin, okay? There's actually 4 different types of opsins when you're looking at each different visual pigment. Let's say, for example, the red one, the blue, and the green for the cones, okay? These opsins are different from all one another, and then also the opsin for this specific one, which is in rods, right? The visual pigment rods, specifically called rhodopsin, is also going to be different, okay? So for the opsins, which is the protein portion, it helps form 4 different types of visual pigments, and that's exactly why I was saying here we have 4 visual pigments that exist in humans: the red, blue, green, and then the one for the rods, which is called the rhodopsin. And I apologize if I'm going too quickly. I think I actually am going too quickly, but I'll try to slow it down maybe. Okay. The cone opsins differ from rod opsins. Okay, so the 3 opsins for the cones, specifically one for the red, the one for the blue, and the one for the green, they're all going to be different from each other. And then also those 3 themselves are also going to be different from the rod opsin, which is specifically the one that's incorporated for this one right here, okay, the rhodopsin. So when we're looking at the protein opsin, when we're looking at the cones specifically, some of these opsins, they make the red cones, okay, and for these visual pigments, they absorb wavelengths of light around 560 nanometers, and that's the reason why they you're able to see the color red, right? Specifically, you're absorbing wavelengths closer to this this wavelength right here. The blue cones will absorb wavelengths at around 420 nanometers, and then the green cones will absorb wavelengths at 530 nanometers. And the thing is, maybe some people might ask, okay, we have the red cones, the blue cones, and the green cones, so how do we see other colors like, let's say, yellow? Well, that happens to do when these cones are working together and they're absorbing wavelengths of, let's say, the red cones are absorbing wavelengths, the green cones are absorbing wavelengths. What happens is there is an overlapping of this information, and then you get your in-between colors, such as, let's say, yellow. Okay, so basically when you see other colors other than the red, blue, and the green, it's really just an overlapping of the different visual pigments from the cones working together and overlapping that information to see different kinds of colors other than the red, blue, and then the green. Okay. Now cone versus rod vision, they have a couple things that are opposite. Okay, for example, the cones, they have a low sensitivity. Why do they have a low sensitivity? It's because they absorb such high wavelengths of light. Okay, so it's higher to basically stimulate them, right? In the dark, they would not get stimulated. Okay, so they're not that sensitive. But looking at the rods, obviously they absorb wavelengths that are at very low levels, so they are highly sensitive. So they are stimulated, for example, in the dark, right? The thing is, though, they react more slowly. If you notice, it's always a little faster to accommodate to going from the dark to the light, versus when, let's say, you're going to sleep and you're trying to accommodate to the dark, it may take about 20 to 30 minutes to basically finally be able to see everything. You know, the normal little peripheral dim vision that you would see, let's say, in the dark, you know. So continuing on, the cones also, though, they react a lot more quickly, though. Versus the rods, they react more slowly. It has to do more so with this process here, what happens when light is absorbed for these guys is they basically become broken down, and it takes a little bit longer for them to come back up. So that's why they react more slowly for the rods. And I'm not going too much into that process because we're going to get into it by basically the next slide. So I'm just going to say that in a more general way right now. Okay, each cone has one type of visual pigment. Okay, we already talked about the red, the blue, and the green, right? So each cone is going to have its own specific visual pigment. One cone has its own direct line to a ganglion cell, okay? And this is not the best picture to look at because each one you see, they all have one direct line. So maybe if I can just jump to the textbook here, the best representation is when we're looking at this over here. So you notice that each cone has its own bipolar cell, right? This cone has its own bipolar cell. This cone has its own bipolar cell. But when we look at the rods, it's a little different. They do something called converging, where you can have many rods, even as much as 100 rods, all converging to basically one bipolar cell, but they will always at least go to the one ganglion cell. Okay, so we have the difference, another difference there, where each cone has its own bipolar cell, therefore it's straight line to the ganglion cell. But we have multiple different rods having, you know, just one bipolar cell, which goes on to its own ganglion cell. So that's another difference for that. Okay. Let's see. Okay, one cone has its own direct line to a ganglion cell. If we look over here, multiple rods, they converge onto a single ganglion, right? They all converge into, let's say, the bipolar cell, which we saw in red, and then the bipolar cell went to one single ganglion. Okay. Now, one cone has its own direct line to the ganglion cell. So connect to a ganglion cell via the bipolar cell. We already saw that here, where that is not shown there, unfortunately. But we basically already saw here that the way that they connect is, in order for the photoreceptor to get to the ganglion cell, they have to have that little intermediate of the bipolar cell. And sorry I keep jumping from textbook to PowerPoint. I know it gets a little annoying, but okay. And also it provides very clear, detailed images for the cones. Only a small portion of the visual field. This is obvious when you look at a more outer picture, for example. Let's say this one. We have this little area here. We talked about the macula lutea and the fovea centralis, where you're going to see that higher concentration of cones. But when you're looking at starting here and wrapping all the way around, and then you reach this area where you have the high concentration of cones, and then it wraps all the way around on the other side, which they're not showing. This is just a sagittal section of the eye. But basically you have all these areas with the high concentration of rods. So basically it's going to be for like the dim peripheral kind of darker processing of vision. But this little small area right here is the area where you're going to have that high concentration of cones. So therefore they don't really get a lot of the actual visual eye field. So for that reason, we have here, they only obtain only a small portion of that visual field because they're just so concentrated on just that one little area. Okay, whether you have the macula lutea, where it's mostly cones and very little rods, and then the fovea centralis, where it's only just cones. Okay. We're just going to skim through this real quick because we already basically discussed this. The rods have a high sensitivity. They react more slowly. Each rod has only one type of visual pigment. Okay, we talked about this visual pigment, which is the rhodopsin. That is the visual pigment for the rods. So the rods only have that one visual pigment. And multiple rods converge onto a single ganglion. We saw how that happens. They converge really to the bipolar cell, and then the bipolar cell go on to the ganglion cell, to be more specific. But because of that, the effects of the rods are summated. So basically, you basically get a summation and addition of all the all the visual stimulus is sent to multiple rods, and because of that, vision is often blurry. Because when you're setting these pathways, right? If you're setting the pathway, this is only 3 here, but let's say this is 20 rods. Okay, so 20 rods are stimulated, and they're sending all the information to one bipolar cell, which is sending their information to this ganglion cell, and then all these are being sent eventually to the brain. Now the thing is, the brain can't basically separate which individual rods are the ones that are more highly stimulated, because it's just getting basically a converging of all their summated visual representations just going to one portion of the brain. So because of that, vision is going to be blurry and not as detailed as when you say the cones will have more detailed vision, because they are able to basically have the distinct separation of understanding which cone was stimulated, because they all have specifically just one cone to one bipolar cell to one ganglion cell, when the same thing does not happen for the rods. So you're going to have more blurry vision when it comes to for the rods versus the cones. Okay, so everything on this slide is going to be already covered on this slide. So I'm just going to go through this slide, and then we're just going to breeze through this one right here. So basically, this is basically the steps that occur when we have our visual pigment here. So for example, the visual pigment is rhodopsin in the dark, and there's going to be 3 steps that we see, in which first we're going to have to make the actual pigment itself. Okay, that's step 1, and then that's in the dark, and then when light becomes absorbed, we actually get a breakdown of the pigment. Okay, that's step 2, and then lastly, we had to regenerate the pigment. We have to remake the pigment, which is step 3. So first, let's cover pigment synthesis, which is just making the pigment, and the pigment we're looking for is, remember, just the rhodopsin. So that's what we're synthesizing here. Okay, just the rhodopsin. So remember that the retinol is derived from vitamin A. So what we have is vitamin A is oxidized, and when it becomes oxidized, it forms the 11-cis retinol. This is one of the isomers that we were talking about, right? And when the 11-cis retinol binds to opsin, it is now called the visual pigment rhodopsin. And that's basically it for step 1. We now have our visual pigment. And notice that the visual pigment for 11-cis retinol here has a more bent shape, okay, specific to basically this isomer for 11-cis retinol. Now, what happens when a photon of light is absorbed? What happens when finally light energy is absorbed? Well, that's when we get something called pigment bleaching. And the bleaching itself really just means the breakdown of the pigment. So we're just breaking down the pigment of rhodopsin, and we're just separating the retinol from the opsin itself. You can see that they're not bound together. Okay, so when light absorption by rhodopsin, this triggers a rapid series of steps, which we're going to cover in, I think, 2 slides from now, which retinol changes its shape. Okay, we have the more bent shape of this isomer 11-cis retinol. And what happens when it absorbs the light, it basically relaxes this portion of the chain, and then it basically has a more linear version of it, in which now we have the isomer called all-trans retinol. Okay, and when we get this conversion, right, this conformational change of its shape, what happens is that all-trans retinol eventually releases from the opsin. It is no longer bound to the opsin. So we have a breakdown of the pigment, the breakdown of the rhodopsin visual pigment. Now, the thing is, what we need to do after this, though, is we need to regenerate the pigment, right? What pigment are we regenerating? Well, the rhodopsin, right? Now, the way we can do this is the fact that when we're looking at the pigmented layer, okay, so we'll just jump back to here. When we're looking at the pigmented layer, what we're going to find here is there's going to be enzymes located in this pigmented layer. Okay, so each pigment cell is going to have their own enzymes. And these pigment cells, what they're going to do is they're going to slowly convert this all-trans retinol back to the isomer of 11-cis retinol. Okay, and they're going to do it in the pigment cells in the pigmented layer. And keep in mind that this does require energy. So this is an active process. It is not passive, so you need your ATP. And they go back up here, and what happens is you can just continue the same steps once again, where 11-cis retinol, in this case, when we're starting from number 3, is it's already regenerated, so it can just go right on and bind to opsin, and then we have our rhodopsin, which is our pigment synthesis. One thing that they don't have illustrated here is around the top of the illustration they mentioned that you can get 11-cis retinol either from getting the vitamin A and oxidizing it, which is where you actually have to start. But once you end off at step 3 here, you can actually just get 11-cis retinol from the enzymes themselves, and then just continue on to making the pigment rhodopsin. And then it just continues on and on. So I'm sorry if that was a lot, but when we're reading this slide, we're just discussing basically the capturing of light and what occurs. So basically the rods and the cones have similar mechanisms of capturing light, involving the breakdown, right? We talked about the bleaching, the breakdown, and the regeneration, right? Pigment regeneration of the visual pigments. So for example, we use the rhodopsin pigment in the rods. Okay, the vitamin A is oxidized to 11-cis retinol form, combines with the opsin to form the rhodopsin, right? So that's basically the pigment synthesis. The rhodopsin absorbs the light, the bleaching, the breakdown of the pigment occurs, and then we have our all-trans retinols formed, and then the surrounding protein, which is this protein right here, becomes uncoiled. It relaxes, it becomes uncoiled, and we get our all-trans retinol isomer. And then the retinol and the opsin combination breaks apart, right? We get it becomes unbound, and we get our breakdown of the actual pigment itself. For step 3, pigment must be regenerated. So the enzymes convert all-trans retinol back to 11-cis retinol, right? These enzymes in the pigment cells. And then 11-cis retinol is rejoined to opsin to finally regenerate our pigment, which is rhodopsin. Okay, so continuing on for light transduction, we did notice here that we have a rapid series of steps, right? This goes a little bit more into depth from this step to here, okay? And this is basically utilizing something that you guys should have done, I'm assuming already, which is something called the second messenger system, okay? So once again, we're going to have this big slide, but don't worry, we're going to go for this slide first, and then we're just going to breeze through the last one. So basically, this mechanism is using the second messenger system, where we have our visual pigment here, which is our retinol. In this case, it's 11-cis retinol, and it's bound to our protein portion, which is the opsin. And what happens is when the light comes in, and the visual pigment, specifically what part of the visual pigment, the 11-cis retinol. Remember, that's the light absorbing molecule. When it absorbs the photon, when it absorbs the light, right, what we happen is, remember the conformational change, that changes its bent shape to the all-trans retinol. Okay, but when the light is absorbed, this actually activates the pigment. So what happens when the visual pigment is activated? Well, then it activates something called a G protein. This G protein has a specific name called transducin. Okay. So what does transducin do? What does this G protein do once it becomes activated? Well, it goes and looks for an effector enzyme called phosphodiesterase. When it finds phosphodiesterase, it's going to bind to phosphodiesterase. Therefore, it's going to activate it. Okay. And phosphodiesterase is something called an effector enzyme, in which he's responsible for converting a certain molecule to another molecule. And first, before we see what he converts to another molecule, First, let's see what it's responsible for in the first place. So here we have cyclic GMP, and what happens is in the dark, they're mentioning here, in the dark, this whole mechanism from here all the way to the left does not occur, okay, in the dark, because when light becomes absorbed, that's when we get the breakdown of the pigment itself. Okay, so if there's no light, everything from right here in left is not occurring. So in the dark, usually these protein channels for cations, specifically let's say sodium and calcium, are open, right? What do sodium and calcium do? Well, they're positive ions, and when they come into the cell, they depolarize the cell. Okay, so in the dark, the cell in this example is the photoreceptor. The photoreceptor will become depolarized because these channels are normally open, because this cyclic GMP is normally bound to these cation channels while in the dark. So that's what occurs in the dark. Now, what happens in the light? Well, in the light, this whole mechanism becomes activated, right? We have our visual pigment becomes activated because of the light being absorbed. We get our conformational change from the 11-cis retinol to the all-trans retinol. We get the activation of this transducin G protein. This activation will go and look for this effector enzyme, which is called phosphodiesterase. When it binds, it will become activated, and now it starts doing its job, right? It takes the cyclic GMPs and basically converts them to just GMP. Now, if we convert cyclic GMPs to GMP, that's only going to make cyclic GMP levels fall. Now, if those levels decrease, then less of these cyclic GMPs will be bound to these proteins. So because of that, with this whole mechanism in the light, what happens in the light is we get these gated channels here that normally cyclic GMP would be bound to are now closed. So if they are closed, we will get no depolarization for this cell when in the light. Okay, so basically in this case, the cell would actually become, which is we're looking at a rod here, for example. This will become hyperpolarized, right? If it is not depolarized, if the positive ions cannot come in to depolarize, then it would just become a little bit more negative. Let's say going from negative 40 millivolts to, let's say, negative 70 millivolts. And don't memorize the numbers. We're just going to say the channel's close, therefore the cell becomes more hyperpolarized. So and this is just light transduction. Okay, we're just converting the light energy into electrical energy here. So the light receptors depolarize in the dark, and then they hyperpolarize in the light, which is what we discussed with these two examples, right? In the dark, positive ions can come in, therefore they come depolarized in the dark. And you can even use, I guess, the D from the dark and the D from the depolarized for memorization purpose. In the dark, they depolarize, D with the D, right? And then the light, these channels are closed because the cyclic GMP levels fall. Therefore, we can't have the positive ions coming in. Therefore, they hyperpolarize while in the light. Exposure to light causes a certain action channels to close. So in the dark, these channels are always open, which we already saw that in the last slide. For example, when we're looking at the visual pigment rhodopsin, light activated rhodopsin activates the G protein, which we saw on the other slide. The G protein binds to the effector enzyme, right, PDE, phosphodiesterase. Phosphodiesterase breaks down the cyclic GMP to GMP. And in the dark, normally, cyclic GMP is bound to these channels, keeping them open. And then the sodium and the calcium can enter freely, therefore depolarizing the cell to around negative 40 millivolts. Now, in the light, though, the cyclic GMP is not present due to conversion by the PDE, right? They convert the cyclic GMP to the GMP. Okay, so therefore the levels fall, and usually the cation channels will close in the light. So sodium and calcium cannot pass, and the cell will hyperpolarize to around -70 millivolts, more negative. And I'm sorry I kind of just ran through this slide, but it's really all we already discussed from this exact slide. So really this would be the slide I would want to concentrate on, and then it basically just tells the story about what this slide is telling me. This is the slide I would want to concentrate on for this, and then this entire slide is basically summarized within that. For me, it's always easier to tell the story using the picture versus telling the story with the words. I would always start with the illustration first. Continuing on, information processing on the retina. So we have the photoreceptors and the bipolar cells generate the graded potentials only. So we already discussed that, how those only generate graded potentials, and then the ganglion themselves are the ones that generate the action potentials. Okay, so for example, the photoreceptors, they do make the graded potentials, which are the receptor potentials, and then the bipolar cells, when they make their graded potentials, depending on the bipolar cell, their postsynaptic potential, their PSP, is either going to be inhibitory or it's going to be excitatory. It just depends on which bipolar cell it is. These things, they always get a little bit more specific. They don't cover more into the details into what the bipolar cells do as much in terms of inhibitory and excitatory. There's always going to be a higher explanation in terms of other classes. For example, for this, we're just going to be responsive for the bipolar cells. Either they do an inhibitory postsynaptic potential or just an excitatory postsynaptic potential. So now, for the inhibitory postsynaptic potential, what neurotransmitter do they use? So if you guys learn about glutamate, glutamate is normally seen at synapses that are inhibitory. It's not always the case, but it's more so common to see glutamate as an inhibitory neurotransmitter. And the thing is, the glutamate is actually not released when light is present. Okay, and all this is going to be explained in another slide, specifically this one right here. Okay, so it's saying in the presence of light, glutamate is not released. Alright, so we have our light here. Right, we're going to explain this exact slide. I just want to show that the glutamate is here. And we learned that in the light, this rod becomes hyperpolarized. So because he's hyperpolarized, he doesn't release his, he doesn't release his neurotransmitter onto the bipolar cell. So glutamate is not released in the light. And we see the opposite effect when it's in the dark. Remember when you said D with the D, right? In the dark, it becomes depolarized. Okay, so when he becomes depolarized in the dark, he does release his neurotransmitter, which is glutamate. So the bipolar cells depolarized, release neurotransmitter onto the ganglia cell. You know what? I really want to just go to this one right here, because this would explain specifically, let's see, if this is the light, this would be this one. And action potential is transmitted to the brain. Yeah, let's cover these two slides, and that would explain exactly what's occurring right here. So let's look at information processing on the retina, specifically when in the dark. So we kind of already talked about this first part. We talked about the second messenger system, right? In which in the dark, the photoreceptor becomes depolarized, right? Remember the ion channels are open in the dark, right? We can see that here. So in the dark, these channels are open. Positive ions are able to come in and become depolarized. Now, if the rod becomes depolarized, right, then he's going to send his little graded potential down along, and then he's going to be able to open these voltage gated channels over here, the calcium specifically. If calcium comes in, he's able to release his neurotransmitter, which is glutamate. Okay, so inhibitory neurotransmitter glutamate is released at the photoreceptor bipolar cell synapse. So this is the synapse at that level. And remember, the glutamate is inhibitory. So this will hyperpolarize the bipolar cell. Now, if he becomes hyperpolarized, he himself will not release his neurotransmitter. Okay? So this will prevent the neurotransmitter release at the bipolar ganglia synapse, this is the synapse now we're talking about. Therefore, there will be no EPSP, no excitatory postsynaptic potential at this level, and therefore the ganglion cell will not be stimulated. If it is not stimulated, there's no information that's going to be sent to the brain. Okay, so no neurotransmitter release means no action potential is generated at the ganglion cell. And this would make sense because since there's no light here, there's nothing really to send for, let's say interpretation of a bright light, red, green, blue, things like that. Now the opposite happens when in the light, right? In the light, we said that the rods or the photoreceptors become hyperpolarized, right? In the light, you have this entire mechanism occurring when in the light that eventually closes the channels. Therefore, if it closes the channels, these positive ions cannot come in. Therefore, the cell becomes hyperpolarized. So what happens when it becomes hyperpolarized? Well, if he's hyperpolarized, he will not have these channels in the synaptic terminal open. Therefore, he's not going to release his inhibitory glutamate. Okay, so what happens is the inhibitory glutamate is not released. Well, what happens with the bipolar cell is now, instead of becoming hyperpolarized, with the lack of this inhibitory glutamate, he actually becomes the opposite. He becomes depolarized in the absence of the inhibitory glutamate. So if he becomes depolarized, then he himself will be able to open his channels at the end. Right, the calcium can come in, causing the release of his neurotransmitters, which will cause the excitatory postsynaptic potential in the ganglion cell. And remember, we said that only graded potentials occur at this level. Okay, so from the photoreceptors that have the photoreceptor potential, and then the bipolar cells that have the graded potentials, they only do the graded potentials, and it makes sense because the action potentials are for long distances, right? The bipolar cell does not have that long distance to deal with, so graded potentials work just fine for the bipolar cell, and the same thing works for the photoreceptor. But the ganglion cells, they have a long way to go, right? Once they reach the optic nerves, they get to a certain portion of the brain, which then sends it all the way to a certain portion of the cortex. Okay, so they have a lot longer area, so action potentials are necessary to be generated at the ganglion cell level. Okay, so let's just photoreceptor cell hyperpolarized, the glutamate no longer is released at the photoreceptor bipolar cell synapse. The bipolar cell depolarizes in the absence of inhibitory neurotransmitter, and depolarization causes neurotransmitter release at the bipolar cell ganglion cell synapse, causing an EPSP for the ganglion cell, and you get your action potential to be propagated across. So with these two slides here, this really just makes sense now that the bipolar cell is depolarized in the presence of the light. They release the neurotransmitter onto the ganglion cells, and then the ganglion cells propagate the action potential to the brain, right? But basically, when they propagate the action potential, it's first transmitted through the brain, via these nerve fibers called the optic nerves, which you guys did in lab. So what about light and dark adaptation, right? How do we adapt when going from, let's say, a dark room and going into a very bright room, or also vice versa, going from a very bright room to pitch black dark, right? How does our photoreceptors basically try to adapt to that? So we have our light adaptation and then our dark adaptation. So for the light adaptation is moving from a dark room to a light room. This is accompanied by an inability to see clearly, right? You get this very glary, quick vision white light, right? And it's because we get that very glary white light vision is because the visual pigments, right, the bleaching of the pigments they break down almost immediately once they have all this light energy, right, being absorbed. When that happens, the cones become stimulated, and then the rods become stimulated in order to break down that pigment when they absorb those photons, the light energy. So you get a flood of signals, results in that large glare in vision that you would see when going from a dark room to a light room. So what kind of adjustments can we make to make up for that, to basically accommodate for that, to adapt to that kind of scenario? Well, the rod's function will basically be turned off. And remember, the rods are very highly sensitive. So if you turn off the rods, you lose that retinal sensitivity. And then, therefore, the cones system will be activated. And remember, these are much less sensitive because they only absorb higher wavelengths of light. Right? Bright colors are higher wavelengths of light. They're much less sensitive. And basically, you get from here a higher resolution. You get visual acuity, which is basically a higher resolution, more detailed image, therefore, because of this. And remember, we said with the rods, it's more blurry vision because of how they converge to just basically reach one bipolar cell and then to go to the ganglion cell. So for the dark adaptation, it's really just the opposite, moving from the light room to the dark room. And we experience this very pitch black experience when we walk into a room like that. And basically the cones just stop functioning. There's no more light energy there to basically maintain the all-trans retinal isomer there and how it separates from the opsin. So the rods are still turned off from the bleaching of the lit room. So since you're going straight from a light room to a dark room, we still have all these rods that are basically turned off from this last scenario here, okay? And because of that, it's going to take a little longer for these for the rhodopsin to basically accumulate. But as it does start to accumulate, which takes a little bit of time, then because they are more sensitive than the cones, you basically regain your retinal sensitivity, right? That increases. And dark adaptation is much slower compared to light adaptation because of this fact that all the rods are turned off, right? Because they were bleached, and they're so sensitive that even one photon of light can basically cause the rhodopsin to break apart. So you need to stay in the dark for longer periods of time away from light energy in order to get that real accumulation of the rhodopsin, because they're so sensitive that they can break apart, they can bleach so easily. So you'll notice it takes about 20 to 30 minutes to really start to see more distinct details in the dark because of this example. Okay, so the dark adaptation is much slower than light adaptation. Now, pathway to the brain. Axons of retinal ganglia are found in the optic nerve. Okay, and this slide does tend to get a little bit confusing for other people, but luckily we have this image right here to help us out. Okay, so the fibers from the medial aspect, okay, of the retina cross over at the optic chiasm. So let's look here. The medial aspect of the retina is here, and the medial aspect of the retina is right here. Specifically, the medial parts, they cross over, medial part cross over at the optic chiasma. Okay, and then they eventually continue on to something called the optic tracts. Okay, so fibers from the medial aspect cross over at the optic chiasma, continue on to the optic tracts, and each eye receives information concerning the lateral aspect of the eye on the same side. Okay, so let's see what that looks like. Okay, so the lateral aspect, we already talked about the medial. From the lateral aspect, we receive the information and it goes to the same side. You can see there's no crossing over from the lateral standpoint. Same thing goes on the other side here. From the lateral, we receive the information on the same side for the lateral portion of the retina. Each eye receives information concerning the lateral aspect of the eye on the same side, information concerning the medial aspect of the eye of the opposite eye, which is what we just looked at, because how they cross over. It carries all the information from the same half of the visual field. So for example, if we have, let's say this is one half of the visual field and then the other half of the visual field, but basically looking at what's just blue. Let's just look at what's blue. We have the medial aspect from this eye and then the lateral aspect from this eye are all responsible for this portion of the visual field, all the blue, right? So this blue and this blue will be complementary to this portion of the visual field. So basically we get this all addition using this medial aspect and the lateral aspect from the other one in order to get the full representation of one portion of the visual field to go to one side of one hemisphere. Okay, so basically all this portion, all the blue will go to this one hemisphere right here, while all the yellow, which is the left side of the visual field, will go to the other side of the brain. The thing is, the lens reverses images, okay? And you'll notice this also when looking in, let's say, a microscope. But they don't have a picture of that here, so I'm just going to jump to this. And I believe it's maybe right here. Okay, there we go. So we're noticing when we look at any kind of light rays, okay, when it crosses over the cornea and then the lens, remember we talked about how the lens and the cornea, they tend to bend the light because it's traveling through a different medium. So we notice that the rays, they bend, and what happens is what we see, what we're obtaining, what the actual light that we're looking at, because of the lens, it actually inverts the image. It actually makes it upside down and then also flipped. We notice that here also, whether it's from far vision or from close vision, the image still gets inverted. Okay. So the thing is, the lens reverses images. So the lateral portion of the eye receives image of the nasal part of the visual field. So let's look at what that looks like. Here we have our lens right there. We have our other lens here. So when we're looking at, I just want to read that one more time. Lateral portion of the eye receives image of the nasal part of the visual field. Okay. So here we have our lateral portion of the eye. And basically, if let's say we follow this, how it crosses over with the lens. Let's choose another area, probably better for this example right here. As we follow this up, we receive information from here. And this is, your nose would be in this area, so this would be the nasal portion of the visual field. So where do we get the actual interpretation of this, the nasal portion of the visual field? Well, that goes to the lateral aspect of the eye, right? And this all has to do with how the lens basically is oriented, in which it receives the image and then it flips the image, basically. So the lateral portion of the eye receives the image of the nasal part of the visual field, and then the opposite will go for the other one. The medial portion of the eye receives the image of the lateral part of the visual field. So we have our medial portion of the eye, and we notice that also you can follow with the colors too. Blue is on this side right here. So we have from the medial portion, if we follow these rays going through the lens, we notice that we have this, the lateral portion or the temporal portion of the visual field is sent to the medial portion of the eye because of the lens. Okay. Now, the only thing I personally disagree with is that each eye gets a complete representation of the visual field of the opposite eye. specifically this portion here, each eye, because when we're looking at here, there's certain portions of the visual field that only the other eye can see. For example, this is the right eye only can basically see this temporal, more lateral portion, and vice versa for the left eye. When going to the textbook, what they say here is going, let's see if I can find it quickly here. Specifically, okay, from here, the left optic tract carries and sends on a complete representation of the right half of the visual field. So it's not the left eye, it's the left optic tract carries a complete representation of the opposite half of the visual field. And the opposite would occur for the right. So if we pay attention to what occurs over here, this is a trend that we see in the optic tracts, where the optic tract will receive a complete representation. If we look at just the blues, which is the blues, this optic tract, which is on the left side, is receiving the right side of the visual field. And then this optic tract, which you see the yellows, which you see this is the other side of the visual field, although it's on the right side, it's receiving the visual field from the complete opposite side. So that might have been some kind of a typo or misinterpretation, but for memorization purposes, I wouldn't go too crazy. I would just memorize that. That's just something that I personally disagreed with, just going through to the textbook. It's really each optic tract gets a complete representation of the visual field of the opposite eye. But if they say each eye gets a complete representation, just go with that, as long as if they didn't fix it in class. Just stick with that. I just wanted to show that it's really the optic tract is really what gets the opposite interpretation, right? This left optic tract gets all the blue, which is the right side of the visual field. Okay, so finishing off, we only have 2 slides. Sorry this took so long, but the optic tracts travel around the hypothalamus. So the hypothalamus would be over here, in which the optic tracts, they travel around the hypothalamus. And the axon synapse with neurons at the lateral geniculate nuclei of the thalamus. Okay, so we see the optic tracts wrap around the hypothalamus and then synapse into the lateral geniculate nucleus of the thalamus, right? We see a lateral geniculate here and then lateral geniculate over here. Now the lateral geniculate nuclei maintain fiber separation, so you have your own distinct right and left fibers separated. You can see that here, where this lateral geniculate, its fibers are not interacting with the fibers of this portion, something we don't see happening at the optic chiasm, where we see a crossing over. So specifically for the lateral geniculate regions, they maintain fiber separation from each other, right? Distinct right and left regions. But even so, they're able to balance and combine their input right from the right side and the left sides, and basically deliver it to the visual cortex. Okay, we can see that from the lateral geniculate, they send that to basically the primary visual cortex, which is in the occipital lobe. Fibers eventually project to the primary visual cortex. Here's where you have your conscious perception of visual images occurs here, right? Which is basically even what's happening right now. You're reading these slides and you're getting a visual representation from the primary visual cortex interpretation. But the thing is, not all the nerves are sent to the visual cortex, right? Some are sent to the midbrain. So we can see that, for example, here, right? So we see that not all fibers are sent to the primary motor visual cortex. Some are sent to the, for example, like the superior colliculus here in the midbrain. And these are just going to help control the extrinsic eye muscles The muscle is responsible for just the reflex. For example, let's say blinking. For example, let's say a fly comes across your face, you have the blinking reflex, right? That's basically because of the midbrain responsible for these visual reflexes, the closing of your eyelids in that reflex. So basically, how is this occurring? Well, or sorry, where is this occurring? Is it comes from a subset of ganglion cells that contain this visual pigment. So we talked about the visual pigments already. This is just another one that very little ganglia cells have. It's called melanopsin. But basically, what happens with these subset of ganglia cells that contain this visual pigment is they're able to directly respond to the light stimuli. Okay, they don't have to receive it via two other cells like we saw with the other ones, with the rods and the cones. So they're specifically able to respond directly to the light stimuli, help mediate the reflexes of the pupils, right? During those reflexes, for example, the closing of the pupil or the dilating of the pupils. And this is located at the pretectal nuclei, which we can see right there. So this, you can have the nuclei in the midbrain, which is the pretectal nuclei, and helps set the daily rhythms at the hypothalamus, basically at the suprachiasmatic nucleus. So once again, we're looking at the hypothalamus area, which we have the suprachiasmatic nucleus. And finally, we're done. Where in terms of depth perception, for example, being able to distinguish, basically having a three-dimensional view of images, right? In order to have that three-dimensional view, basically the depth perception, what's farther away, what's closer, is you basically need both eyes are necessary for this, because the visual cortex fuses the images from the both eyes together, and now you have your depth perception, right? It only works when the two eyes work together. And what they say here is the medial and the lateral eye placement. It depends on how the placement of the eyes. For example, for us, we have medial placement of our eyes, right? If this is the midline, we have more closer towards the medial portion. But let's say something like a pigeon, for example. A pigeon has lateral placement of their eyes. So because the pigeons have the lateral placement of the eye, they do not have depth perception, and basically they have to learn through other learning cues throughout life to basically compensate for that. But for us, we do have depth perception because we have the medial eye placement, and for that we are able to fuse these images together to get depth perception. And just in general, each eye has a field of around 170 degrees. So let's say this is the right eye here, and it goes all the way from right here and goes all the way across to there, which is about 170 degrees. And then vice versa for the other guy. It's going to be 170 degrees. But basically there are portions in which they're both able to interpret those images, and there is some converging happening here. But also remember that that other small percentage is only due to only one eye. Okay, and that basically ends the PowerPoint presentation. I hope that that helped. And if you have any questions, please let me know. And I apologize for how long the PowerPoint ended up being.